Okay. Okay, so today we're going to try to discuss the mycoplasma. And this is part 2 of that series of papers. So this one is hopefully everybody read it. It's the complete chemical synthesis of the Mycoplasma Genitalium genome. Okay, so what did they do? What did they do?
They basically took a whole bunch of smaller parts and constructed into one big part to get like the entire genome. Which
Yes.
is different because they hadn't had like a complete like non-viral genome at the time. So they were like,
Yeah, so okay, you saw that comment. So before this, the biggest synthetic genomic fragment was 32 kb from a virus. And so in this paper, they broke that record by a factor of about greater than 10. So it was 582 kb of the Mycoplasma Genitalium genome. So they broke like a synthetic genome record. Let's see. So it's important to note that this is not yet, so hopefully we'll read at least one more paper in this series. This is not the actual creation of the first synthetic organism. This is just the first, it's like a midway step, the synthesis of its genome. So just keep that in mind. Remember that the Mycoplasma Genitalium, that one is actually a sexually transmitted like UTI. It's a human pathogen. How did they get around that? Like imagine trying to propose an experiment where you're going to synthesize a genome from a human pathogen.
It said that they used something they did to it made it to where it wouldn't take in mammalian cells.
Yeah,
I
so-
remember where it is.
So something they did, you said something they did to it made it so that it wouldn't be uptaken into mammalian cells. What's the something they did to it?
Hang on, I have it highlighted.
Or if you don't know exactly like what we did in theory, what could you do? If you didn't catch
Oh,
that.
they added a defect. Where was it? They added the watermark that added aminoglycoside resistance.
Yeah, so they added an insertion into this gene MG408, which is MSRA. That's the name of the gene. And this insertion into that gene broke the gene. So they essentially made a knockout. Not a complete knockout, but they broke the gene of MG408. And this gene had previously been identified in previous papers of this pathogen. And essentially like this gene is important or essential for its pathogenicity. So they broke that gene so it could no longer be a pathogen. So that's how they sort of, I think, navigated around the fact that this is like a pathogen or can be pathogenic. Okay, so, and maybe this is review, but why do this? Why do this experiment?
it kind of opened the door for like more genetic constructs, I guess, because since they have like a whole genome, like a complete genome, especially one for like maybe this magnitude.
So you're saying like, it was like a technology development in terms of like a hurdle of what can be done?
Mm-hmm.
How do I paraphrase that?
Technological development.
Technological development. Okay, that's good. Technological. Why else do this? In the context of the other paper that we read. So yes, like we're progressively trying to work on this conundrum of how do we synthesize larger and larger genomes so that we can have like designer genomes. That would be a technological development, but why else do this? Like why do it with mycoplasma?
What is life? The essential genes.
Yeah, so that was the other thing is like, what is life? And to really understand life, you should be able to like program it and build a living cell that is self-sufficient. And so that's, again, like that's what they're getting at, is this big question of what is life and what is the minimal genome required for life. Okay, so those are kind of like the big whys. Yes, I agree. Let's just quick review from a little bit more from the last paper. So remember in the last paper there were actually two mycoplasmas that were studied. There was the pneumoniae and the genitalium. What did they do in the last paper with these two mycoplasmas?
Like compared them to see which genes were essential.
Yeah, so they would compare them, and let's say this was the genome of this one, and let's say this was the genome of this one. Which ones are the essential genes? Which category? A, B, or C?
B.
B. B would be the essential genes because it's consistent in both. So they compared these, and there were about 400 of those genes, if you remember. And this research yielded essentially, like in that last paper, then they did the transposon insertions where they shot them in, and they found essentially like 100 of those 400 were non-essential. And so then that's where this paper picks up. And so then obviously what do you want to do in the next paper to see if you can actually like prove this? Like their goal is to find the most minimal genome. So then what should they do?
I think they were trying to figure out what made those non-essential genes non-essential. Or-
They were not
or
studying
not
gene
non-essential.
function in this paper.
They want to like construct all the different genomes with one missing and see like what happens.
Nope, that's what they did in the last paper. So in the last paper, they sort of made all the separate permutations where they deleted each of these genes individually and kind of like saw what happened through the random transposon insertion. How is this paper a little bit different from that, like a next step?
Weren't they, this one, they had overlaps in between and they were trying to connect those overlaps together as well as put in insertions into the essential genes?
Did they put insertions into the essential gene, or the?
I thought they were talking like in one of the images they were putting it like, they had a hot
Connected to these numbers, like, so remember, so I said at the conclusion of the last paper, the conclusion was essentially the genome, the conserved genome between these two is about 400 genes. And of those 400, about 100 of those are not essential, was their hypothesis. But if you remember at the end of the last paper, they were not certain precisely if all those were completely non-essential.
I guess they were trying to rule out false positive non-essential genes. or
So
false
what would you
positive
do?
that
What's the
don't.
next step? To really get to like to prove the minimal genome, like what's the next step?
Put some pieces together.
What?
To put some pieces together of the essential genes and the ones that might not be essential.
Okay, so you're right, you're gonna put some pieces together, but think about like what would you craft as your most minimal genome? What would the genome be?
The ones that are definitely essential and the ones that could possibly take places of the others.
Yeah, so they're in this paper, they're essentially their goal is to assemble like a genome that's like about 300 genes. And so they're gonna test the hypothesis that like, well, first actually they're not testing it yet, but they're gearing up to be able to test the hypothesis of can you have a genome with just the 300 that are essential? Does that make sense? So the difference between this one and the last one is now they're trying to synthesize an actual genome that has every single non-essential gene just deleted, not present in the genome. So they're like removing 100 genes. They're not individually knocking them out anymore. Does that make sense? Okay, so that's how this one is a little bit different. Okay, and if you remember from the last, there was this sort of like conundrum at the end of the last paper, which was like, we've identified these non-essential genes, but is it possible that they can be simultaneously non-essential? Like, is it possible to delete them all at the same time? They hadn't tested that. Okay. Okay, so now's where it starts to get scary. This is actually like kind of hard. I'm gonna do my best here. So the next question is like, okay, how did they do it?
So,
Comments? Yeah.
well, I was gonna say, they basically tried to reduce the genome, so they only use like the like the 300 or so like essential genes.
Right.
Put those into insert those genes to see if they
Right.
supported life.
So their goal is going to be 300, approximately like a 300 or Mycoplasma genome. Yes, I agree, that's their goal. What did they do? What was like step one? Hopefully everybody read this.
They made the in vitro cassettes. They made 4 of them. And they-
Only 4?
That's
Well,
what this
they
says.
made,
In the first stage, they made 4 neighboring cassettes.
For neighboring cassettes, so not 4 total, but 4 neighboring
so
cassettes.
it's like
Of
25
what?
in all.
What?
Oh, I'm saying like 25 in all in like the first build. So it'd be like 4 neighboring cassettes of like,
Okay, so they're essentially making like 4 cassettes like this. What was the approximate like length of the first cassettes? So little, like, so they're assembling, essentially they're assembling small pieces. What's the size of these small pieces?
22 kg.
Wait, what? The smallest,
Oh,
the smallest
okay.
fragments. What's the size of these smallest fragments? About
300? Or
300 what?
no.
Definitely not 300.
Sorry.
Okay, so these pieces are about 7 to 8,000 kb. So 7 to 8 kb. So about 7,000, 8,000 base pairs. Okay, and then that means when you string them together, you're gonna have something about 24 kb, which is I think something like what you said. Okay, so their first step is they're gonna assemble these small pieces. How do they assemble them? How do they get them? What do they do to make them? Those little pieces.
They joined them together.
Before they join them together, how did they make the little pieces?
Says they were individually synthesized.
Yeah, what does that mean?
They like literally just put base pairs together.
How do they do that? Like, did they actually do that?
They ordered it, like, or outsourced it.
Yeah, they literally just like bought them. So they typed them in a computer, told them like what they wanted, and then they had a company synthesize the oligonucleotides that were small oligos, essentially. Like, so my point is they didn't actually build these, they ordered them. So they just typed them in on a computer, told them what they wanted, and then they bought these. Okay, so now they have these pieces. Okay, what's step 2 then?
So they took all of these, they assembled in like step A. So they had like 25 of those for what you grew out basically. And then from those 25, they would take 6.
Step 2 of getting these to come together. How do they get them to come together?
They put them together using bacterial artificial chromosomes.
Okay, so they're using, okay, so that's not how, but that's the holder. So they're using BACs, yes, so they're using bacterial artificial chromosomes.
Is it the in vitro recombination that?
Yeah, so this is interesting. Okay, so they have this word called in vitro recombination. So this is like a weird word. I think this is before the protocol actually got its real name. Is this actually, the way that they did it, is this actually a recombination? Who said no?
Me.
What's actually happening?
They're transformed, or no, not transformed. Because it's not recombination.
It's not recombination.
I'm sorry.
This is the word that they say, like, this is how they quote unquote assembled these into the next larger segment. But my question for you guys is like, what is this? It's actually described in figure 1 part A. So there's a figure that literally looks like this, and then it literally tells you like what's happening.
It's just sticky ends.
What?
Sticky ends or homologous regions.
How do they make the sticky ends? Like they do make sticky ends.
Overlapping base
Restrict-
pairs over there.
So there's an overlap here, there's an overlap here, there's an overlap here. Sticky ends is correct. How do they generate the sticky ends?
The restriction is on.
No, they don't use a restriction enzyme.
I'm thinking about
You cannot, okay, so that like, that's like the whole point of this paper is you cannot use restriction enzymes to assemble giant pieces of DNA. Because as soon as you get bigger and bigger and bigger pieces of DNA, those sites are bound to be in there, and so you start cutting them up. So they do use some restriction enzymes in some certain contexts, which we'll talk about. But the whole point of this is you can't use restriction enzyme cloning to do things like this. So we've actually covered figure 1 part A before in a lecture. That was, like, I've covered that. How do they generate the sticky ends? Yeah, so there's actually, like, they don't have the perfect enzyme for this, I don't think yet, but they have this tack, I think it's a tack that has 3', did you say endo or exo?
I mean exo.
Exonuclease activity. So they're essentially, like, the main point is they're essentially using a 3' exonuclease, okay? So that's gonna chew essentially the ends in like this. And now I actually didn't draw it well, but this fits here, and this fits here, and this fits here. What method did that look like, which I previously described? This exonuclease trick to get sticky ends.
It's the Gibson pattern.
Yeah, this is like, that's how Gibson assembly works. Is Gibson assembly like drawing a blank for everybody, or is that like a aha, I remember that?
Okay, so I was thinking, because like we were using small pieces, I didn't know if it was still considered Gibson cloning.
So this is written by the guy who invented Gibson assembly, and this is probably like in the process of him inventing that approach. Is that, like, that's why this is cool.
Oh, okay, so that's why.
So y'all should read harder. Anyway, okay.
So with the Gibson assembly protocol, it can't, like, that protocol that we learned in lecture, it can't do this kind of thing, right? Like, you
Why
couldn't
can't-
make, I thought there was a limit on the,
6, I
That's
think.
like 6
Yeah.
But they're only assembling 4.
okay.
Okay, so this is essentially Gibson assembly. So this weird like in vitro recombination, this is not recombination. I don't know why they picked that word. Maybe there's some like historical context, but this is essentially like a primordial Gibson assembly. Okay. Okay, so once they are, so step 2 is like Gibson assembly. And now you have essentially something that looks like this. Okay. And then what happens? What's the next thing that happens? So now the length of this thing is about like 24 kb. What do they do with this thing?
They take the 24 kbs, they take 3 of those and put them into.
Wait, so first, this is just a linear thing of DNA. This thing that they just assembled. Is that stable? Who said no? So yeah, so how do they make it stable?
Is this when they put it into the bag?
Yeah, so now they clone it into a bag. How do they clone it into the bag? This is essentially figure 1 part B, C.
They're not using, I thought they were using it in a E. coli stuff.
So in E. coli, yes, clone it into a bag in E. coli. My question is, how do they get this into the bag?
PCR.
What?
PCR.
How, wait, so you're, yeah, kind of. How, what do they
use
PCR?
cloning primers, and then they use like sticky ends on the primers to.
On this?
Yeah.
That's definitely not, so let's actually think about that. So if they did some kind of like a weird like cloning primer thing where they put restriction enzymes here, what's the problem with doing that? How big is this fragment?
Huge.
It's giant. So they want to do this without mutating anything. So at this point, like, they can't use PCR on this thing.
Yeah.
But you're right, they actually do use PCR. What do they PCR? Look at figure 1, B, and C. Primer 66 and 69.
So they make more sticky ends, or like they add the back sequence onto the artificial chromosome?
So they do. I don't know what you mean. Okay, so there's this. So let me
Okay, so.
erase this, and let me actually like draw the. Hopefully everybody's got this. Let me actually draw that part of the figure. So here they have. So this is in figure 1B. They have something that looks like this.
You know this whole time.
They have something that looks like this. What is this showing you?
The primer design?
Yeah, so, okay, so so these are their primers, primer 66 and primer 69. And what are they amplifying? This. So they're PCRing the back, not their Gibson assembled like large chunk. So they're PCRing the back, okay? So they essentially create an amplicon that looks like this. So imagine this is now, it's not circular anymore, it's linearized, okay? And if you look closely at the primers, there's a not one site and a not one site. And then this is essentially like homology. Call this left, call this homology right. This one matches here, that one matches here. And so now how do they get them together? So finish the number 3. How do they get this to join to that? Is that a trick?
You just mix them.
Well, if you just mix them, so there's actually no overhangs at this point. Like this molecule is double-stranded. So it looks like that, and this one is double-stranded. So they can't come together if you just mix them. What needs to be done before you mix them? Just like in the very beginning, when I drew this, you can't just mix these two together. They won't like come together. What do they have to do to get these to come together in step 1? The exonuclease. So what do you think they should add here now? Yeah, so they'll add an exonuclease. Okay, the exonuclease is now going to chew in, and it'll, I don't know exactly, whatever, something like that, chew in. And now this can match that, and this can match that, and then it circularizes. So that's what the little drawing here in C, that's this process circularizing. That's how they get it circularized. Okay.
What are this number 67 and 68 on figure C?
What do those numbers mean?
Are those more prime? Because they named.
Those are just like sequence. I think those are like 66, 67, 68, 69. I think those are just segments of the genome. Like I think it's like an example, 66, 67.
It was like.
Yeah, they're paired together in 4.
And they just use it as an example.
Yeah. Okay. So we understand now how they go from, in step 1, they have these 7, 8 kb fragments. We understand how they come together with essentially a Gibson assembly into a 24 kind of kb size plasmid. And then they actually PCR the backbone of the back with special primers.
So is this when they use the T4 polymerase? Because at first I know I said use the T4 polymerase and then the tag, and I know the T4 has 3.
I think they're using some polymerase that has an exonuclease activity.
Cause that's what I was at. Cause I know like T4 has 3' and 5' exonuclease activity, like in the absence of the DNTPs.
The important thing to remember is that it's an exonuclease. Like that's
So like,
all you need to remember.
okay. Well, I guess my question would be, how do you know if you need to use one that has a 3' and 5' exonuclease versus just one that has like either or?
If it was an exonuclease that was cleaving at both 3 and 5, why would
so
that
it
not
just
work? Wait, why would that not work?
Oh, because it's gonna cleave all.
It's just going to chew it in like uniformly, and there's going to be no sticky ends. So it has to be either one or the other to create sticky ends. Does that make sense? Okay. Okay, so now they've assembled, so let's go 3. So now they've assembled this thing that looks like this. They've assembled like this thing, okay? And actually, this part would be much bigger. So I drew this, if I was to draw this like accurate, like size-wise, it'd be something like this. Okay, so they have this. This is still not the assembled genome. So this is the input for step 4. What is step 4 now? This is where it starts to get hard.
Is this when they electrophorated into E. coli and TH10?
Well, okay, so yeah, so at every sort of like completed assembly step, you have to put it back into E. coli. They would freeze it as like a glycerol stock, and then they would essentially be able to mini prep from the back to collect it. So yes, that's correct. But my thought is step 4 is sort of like, okay, we understand how this construct was made. Now how do they go to the next level construct? So we've essentially only seen the first synthesis step, which was take the little fragments to generate the larger fragment, which then got inserted into the back, which is that. How do you go to the next level of assembly at a larger level?
Was this
You cleaved it?
Yeah, so this is actually now where they are using restriction enzyme. So they cut with not one. So imagine they had 2 of these. So they did this process twice, except they have a E, F, G, H, not one. Okay. So they have 2 of these, and now they're going to the next process of assembling to the next level. So they cut with not one in separate reactions. So they'll cut this, that'll essentially open up that. Then they'll cut this, that will open up that. I should have left the sites. Okay. And there's these not one sites. Now the next question is, how do they get it? So what they want to get is A, B, C, D, E, F, G, H. They want to get from these to that. And so this was what?
Oh, I was gonna say they formed it into that ectoplasm that you drew at first.
How did they do that?
Well, after they cleaved it with the not
So they cleave it with not one, yeah, that's the first part. Then what? It's not anything new.
It's the they digest it.
Well, they already digested it. You already said that. So if you just digest these, what's missing? They don't actually ever use like a ligation, I don't think. But what's missing now so you've digested, so they essentially they repeat that PCR process. So they have they have that PCC, whatever it was, something like that. Okay. They repeat that process with these special primers. They PCR this. Let's make that product like green. So they PCR that. And when they built this, they built I should use a different color now. Because now it starts to get real complicated. They built an overlap here that matches here when they built this. So they originally thought this through, okay? And now so if this is for I, double I would be PCR, the backbone, with those special primers again. And then they do the exonuclease again. okay? And if you look at, where is it? Figure 5. So look at figure 5. This is kind of magical, like I don't know why this happened, but essentially like the interesting thing about the next process is the not one sites get removed in the next synthesis. Like when the T5, or I don't know if it's T5, but when the exonuclease is chewing, yeah, when it's chewing back these sides, it messes with the not one site, and so it essentially like messes up the not one site and removes it. So now these not one sites are like removed. That's what this figure is showing. Like here they have not one, and then after it chews in, it's removing the other part of that not one site, which messes up and gets rid of the not one site. And then then it comes together, okay? And then it comes together, and they're actually adding in new not one sites from those primers on the backbone of on that back. So this is what's really interesting about this process is every time, and then they repeat this, like every time they assemble a larger fragment, they cut it with not one, the exonuclease removes the old not one sites, and then they add in new not one sites with these special primers that have them built in. So every time they generate something bigger, they get essentially like another plasmid that can be linearized at the same spot with these not one sites. Like it's kind of hard to understand, it's even hard for me to understand, but if you look at this figure 5, that it's showing you like that's what's happening. Any questions on that? Or does that kind of make sense? Like the reason they have to remove the not one sites, what if they left them in? If they left in the not one sites, the construct would then look like this. And why would that be a problem?
Because
It's in the middle of like the piece that they're trying to see.
Yeah, so if they did not remove the not one sites, and then they tried to go to the next step to assemble like the next two pieces, it's just gonna cut them back down to the original size. So this has to get removed. Those sites that were originally on the ends have to get removed. They get removed, and then they add them back into those primers so that they're always coming back in right at the border of their larger construct. And so now they can just repeat this process to assemble larger and larger fragments. And they probably would have just kept doing this to assemble the entire genome if what wouldn't have gone wrong, what went wrong at some point.
once they got to like trying to assemble like the C and D parts, the D and E parts, they realized it was like too large. I think
Yeah.
the issue
It was too large for what?
for
Yeah,
the
so essentially like at some point, like they keep doing this, and at some point it gets too large. And E. coli does not like the pulling this stuff in the back anymore. So it essentially like starts to break down, it's not working anymore. So then how do they fix that? What do they go to next?
yeast.
Yeah, so then they switch to YACs, which are yeast artificial chromosomes, okay? And in the classical sense, yeast artificial chromosomes have a centromere and autonomous replicating sequence, so it's kind of like an origin, and they have telomeres. And so those would be linear chromosomes, but that's not what they use here. Here they use something a little bit different. How is it different? They did use a YAC, but how is it different? Different from a linear chromosome?
Are you saying how?
How is the YAC that they use different from a linear chromosomal YAC? Yeah, it's circular. So it doesn't have telomeres. So essentially, like the thing they have is essentially just like another plasmid, except in yeast, it's a YAC. And yeast can handle much larger fragments. So essentially, like they get to the limits of what can be done in E. coli size-wise, and then they switched over to yeast, and they start using a YAC system, okay? And they switch their cloning methodology to, they switch their cloning methodology to essentially this technique called TAR, which is transformation associated recombination. Here, recombination is actually what's happening, okay? And transformation associated, that just means that they're transforming in linear fragments, and once they get inside the yeast will recombine them to create a circular yak. Why does yeast do that? Yeast is just really good at recombination. Like, it just works well in yeast. And so if you look at figure, no, this is I think the final figure, one of them. Yes, figure 6. They have something that looks like this. Like, I'm not getting it perfectly right. Like this. They have something that looks like this, where imagine this was larger section, let's call this like, what do they call it? Oh, they call them C. Let's call this like C1, C2, C3, C4, C5, and then this is the yak backbone.
Wait, Dr. Beckmann,
What?
didn't they combine the back and the yak backbone?
The yak and the back, like the yak is
Right.
built so that it has two features. Like, it can be treated as a back or a yak.
Oh,
It's essentially like it's got
But
the
it's
stuff
the-
to make it a back, plus it's got the stuff to make it a yak. So you can like,
I didn't
Why
know,
would they keep it?
because
What?
they broke it into 2 pieces.
why'd they keep what?
The back cassette or any part of it that can't survive through E. coli anymore?
I don't know if it can't survive in E. coli. It's like,
Or like, it'll-
it's probably that it wasn't assembled well in E. coli. But maybe that's just what the plasmid had. I don't know if they're ever transferring it back into E. coli after this point. I don't know. But it would be a lot harder to just like this is something probably that they had in their database that worked. And so it's like, well, let's try with this. And it worked. As opposed to like, well, let's delete this, and if that makes sense.
Yeah.
Did you have another question?
It was kind of like piggybacking off that.
Okay.
I was just like,
Okay, so this, how is this now assembly process subtly different from what they were doing in 4? Not you. How is this step 5 different from what they were doing in step 4?
It's like actually doing recombination.
Yeah, this is actually recombination, and so you don't need to add any like T5 or exonuclease stuff. Like, this is actually recombine. As long as you have the homology arms, the yeast just recombines it. Like, the yeast just does it for you, and it recombines it, and it creates an assembly. Okay. So that's how it's subtly different from these, where before they were adding these like exonucleases to get these tricks to like assemble. Now the yeast is just like doing it.
When it comes to choosing between doing something like this in yeast or E. coli, what would be the benefit of using E. coli if yeast is so efficient at recombination?
So that's a really good question. I was actually literally just thinking that is like, well, why didn't they just use yeast the whole time? The benefit of using E. coli is that the doubling time is much faster, and so you could get an experiment done and see the result the next day. Yeast, you'd have to wait 3 days. So in terms of like speed, they probably like, I don't know. I don't know the actual context of this, but if I was the researcher, they would probably like, let's just start in E. coli. They probably just started there because it was faster, and they probably did not think that E. coli would reach the limit. Like, maybe they thought they could assemble the whole thing in E. coli. And then once they hit the limit, they were probably like, well, let's try something else.
Is E. coli cheaper, like in any significant way, when you're dealing with cultures and growth and stuff like that?
That's an interesting question. They're both considered like cheap in terms of like science-wise, but time is the most valuable thing. And so I would say yes, E. coli is cheaper because an experiment that takes 1 day takes 3 days in yeast, unless it's not working. But yeah, and it's probably like a hindsight is 2020. Like, maybe if they went back, maybe they would just do the whole thing in yeast. I don't know for sure. Okay, so they get that assembly. Okay, let me see if I did not cover everything. So throughout this entire process, as they're like assembling these things, how are they checking whether the assembly worked or not?
Earlier, when they were still using E. coli, they were using field
Yeah.
inversion gel electrophoresis, and then here,
Yeah, so okay, so wait, I wanted to talk about that. So In their paper, they said field inversion gel electrophoresis. This is essentially like the same thing, I think, as pulse field gel electrophoresis. So we know what gel electrophoresis is, right? You make an agarose gel, separates stuff according to size. What's the problem with standard agarose gels in this paper? Why couldn't they just run a normal agarose gel? Emma. What's the limitation of a normal agarose gel that makes it not work in this particular case? Chris?
Size?
Oh, yeah, okay, so, yeah, okay, you got it, right, yeah. So the size, what's the, when you run a normal agarose gel, like what are the sizes of things you're dealing with?
The supercoil.
What are the sizes of things that you're typically running in a normal agarose gel?
Do you remember the ladder size that we used?
You're right, you're right, like the limitation is the size of the things that they're making. So
Like this one's too small, it's too big.
yeah, this agarose gel only deals with small stuff. It's essentially like, you can run things that are like 100 to probably like 10 kb. Anything bigger than 10 kb, what is, if you ran a 30 kb thing on this, what would you, let's say you were trying to compare 30 versus 35 kb, and you ran those on these lanes, what would the gel look like? They'd essentially just be like this. Like you would not be able to tell the difference because these are so big they can't like get through the gel. Okay, so like this is a common thing in molecular biology where you kind of like learn like the base way to do something, but then you realize there's a whole bunch of different like modifications to the protocol that you can do to make it work with other things. So in this case, the sizes of the things that they're working with are not, they're much, much greater than 10 kb. Like they're dealing with things that are like at the very smallest, almost like 10 kb, up to like 100 kb, and at the end, like millions of base pairs, okay? So they go to a different technique, which is a subtle modification of gel electrophoresis. So pulse field gel electrophoresis is essentially like a different apparatus where you have an electrode here, let's see, so the DNA is negative. So essentially like pulse field gel electrophoresis, it will temporarily like shock it to make it go this way, and then it will shut that current off, and then it will temporarily shock it to go this way. So it's essentially like making it run in like a zigzag pattern, okay? And the reason that that separates things out is like if you have like some gel matrix.
Oh, this is a good one to think about.
What?
That's like a cool drawing, cause I understand.
Well, if this is like a gel matrix and the DNA has to like go through these, right? Essentially like when you're changing the direction, it just enhances the effect where smaller stuff, like if you're constantly changing the direction, smaller stuff is able to get through faster, and gigantic stuff goes much, much slower. So it's essentially like to get, it's a way to get a better resolution on gigantic things that are running through the gel, okay? So they do essentially field inversion. I think it's just another form of this pulse gel. It's essentially like they're just changing the direction of the current temporarily.
Would it be a problem for like SDS gels too? Or is the same, like protein thing too big?
Like, is there like a pulse field SDS? I don't know, you'd have to Google that. I think proteins, like typically the protein size ranges that you're dealing with, it's not, proteins are not typically like DNA, where DNA can go from like tiny to just millions. There's no proteins that have millions of amino acids. So essentially like the biggest proteins are like there's obviously going to be giant proteins above this that are like rare cases, but like typically your biggest proteins are like 250, not kb, kilodaltons.
So when they do like, well, I guess when they're studying like holo enzymes or multiple proteins that form a complex, they would separate in the SDS page.
Well, you can do, in that case, you can do non-denaturing SDS page, where you can electrophorese things that are in complex, but you can't add, it's a non-denaturing gel, you can't add SDS. So yeah, there's definitely like subtle modifications to page as well for proteins, although it's gonna be a little bit different because most of the protein size-wise can separate out in a normal page. Unless, yeah, you may be running complexes or something. Okay, so that was the pulse field gel electrophoresis. What time is it? Okay, I got 5 minutes. Is there anything else that I did not cover? Or did anybody have any questions? So what's the next step? Like where did this, so this ended with essentially like, what's the end point here? Tell me like where they ended. Describe to me the situation where the paper ended. Chris.
Didn't they like, they were like, in their conclusion, they were talking about how only like a quarter of it, they could only see, like get a quarter of it, because if they did half, it'd be too big.
Wait, what? I think they got the whole thing.
They did eventually, but I thought when they were talking about it, they could only, like, the reason why they had to switch from,
Oh yeah, the bacteria, E. coli sort of like gave out at like a quarter of the way there or something like that. Or maybe it was halfway there. And you're right, they switched to yeast. But they eventually got the full thing. So what's the end point?
to have their whole,
Yeah, so at the end point is essentially like they have like a yeast cell with the entire Mycoplasma genome stably replicating in the yeast cell. So that's the end point. So then my next question is, if that's the end point, what's the next step for their quest to synthesize a minimal cell?
Just sequencing or
They did sequence it, so like they definitely, that was like a part of the process. They wanted to make sure that they weren't encoding errors. So they sequenced it like multiple times throughout as they're assembling. But that's not the next step.
oh, do you mean like with the next paper?
Like what's the next step to create, is this the minimal cell?
Oh.
Wait, is that the minimal cell?
No,
Why?
because it still has that Beckmann. Yeah.
No, like I mean, that's not why.
Cause it's a knee.
Yeah,
no,
this is
okay.
not the minimal cell, because this is yeast. Like, yeast has its own chromosome. Like, this is not an autonomously, well, it is an autonomously replicating thing, but
But it's not an E. coli.
this genome is not really doing anything in yeast. It's not being encoded. It's not like, it might have been transcribed a little bit. Like, I don't know for sure. But how do they get from this to the final step? What do you think? What should they do?
Have to somehow put it into an empty cell.
Yeah, so you somehow have to figure out a way to take the synthesized genome out of the yeast and then somehow get it into like a mycoplasma cell, an empty cell. So there's going to be probably in the next paper, again, like I'm actually going through this as you are going through it. I haven't read these papers before. In the next paper, they probably have to figure out like how to remove the genome
From yeast.
from mycoplasma, not yeast.
Oh, sorry.
From mycoplasma, like that. This is just going to be like they're going to prep the plasmid.
Oh, okay.
Like they'll grow a bunch of yeast, they'll purify a bunch of plasmid, but they have to figure out how to remove the wild type genome from a mycoplasma to create an empty cell without killing it. Like you create an empty cell, and then number two, you have to figure out how do you get that into there. So then there would be like some type of weird transformation into there, and then you hope that this thing now replicates and starts its own colony. This is, I think, the next part of this paper, or the next part of this series. Does that make sense? Okay, we can stop it.